Bom dia a todos. Espero que vocês estejam todos bem. Sejam muito bem-vindos a essa quarta edição do Simpósio de Ecofisiologia Sistêmica. É uma edição muito especial, não só pelos tempos de pandemia, mas é uma edição que, de certa forma, representa uma espécie de resistência para a gente. Apesar de tudo que está acontecendo, a gente resolveu realizar essa edição, e é um evento que ele traz no seu bojo, a ideia da gente estar discutindo, questões que não fazem parte necessariamente do nosso dia a dia, Então, é uma forma da gente discutir as fronteiras da ciência, discutir questões que normalmente não são trazidas, nas nossas pesquisas e que fazem parte de uma nova perspectiva da ciência. Então, para nós darmos início ao nosso simpósio, eu vou convidar aqui os membros que vão compor a nossa mesa, Primeiramente, o professor Danilo Daloso, representando a diretoria da Sociedade Brasileira de Fisiologia Vegetal, o professor Rafael Ribeiro, como membro do comitê organizador e também representando aqui a nossa revista da sociedade, a Brasília Theoretical Experimental Plant Physiology, e o nosso palestrante especialíssimo, professor Ulrich Lüttge. Então, bem-vindos todos vocês, é uma grande alegria recebê-los aqui, e eu passo a palavra, sem mais delongas, para o Danilo, das boas-vindas em nome da nossa sociedade.
Obrigado, Gustavo. Obrigado a todos os organizadores do quarto SES. Essa ideia nasceu de uma reunião de um dos grupos aqui de fisiologia, e a gente está feliz que se concretizou. E em nome da sociedade, a gente tem um prazer enorme de dar o suporte para esse evento, dar o apoio para esse evento. Não só esse, eu acho que esse ano é um ano bem especial para a fisiologia vegetal do Brasil, porque a gente está, como o Gustavo falou, nessa situação de crise da pandemia. Nós, fisiologistas, nos organizamos e fizemos vários eventos. Então, esses eventos foram todos feitos por associados da SBFV, e a gente, como membro da diretoria, tem o orgulho de apresentar mais um evento com uma programação excelente. A gente já acabou de ter o Simpósio Brasileiro de Fotossíntese, o primeiro simpósio, que foi online também, um sucesso, e ainda teremos mais 3 eventos, ou no mínimo, 3 eventos até o que a gente sabe, tem mais um sendo confirmado, então mais 4 eventos daqui até dezembro, sobre fisiologia vegetal com o suporte da diretoria. Então, é um ano muito especial, e para coroar, esse ano a gente tem essa palestra de abertura que vai ser, com certeza, sensacional. E também aproveitando para divulgar o nosso congresso da sociedade, que será o ano que vem, que o Gustavo é um dos organizadores, já vai acontecer. A gente espera que presencialmente, a gente espera que a pandemia até lá nos dê uma trégua. Então, o congresso está organizado para ser em setembro do ano que vem, de 19 a 23, em Porto Alegre, no centro de eventos da PUC. Também uma programação já quase que completa, vocês podem checar no site, muito já avançada, com toda a programação, todos os palestrantes confirmados, enfim, a gente está trabalhando duro, e o Gustavo e o pessoal de Pelotas, à frente aí dessa organização, tem feito um trabalho magnífico, Então, sem mais delongas, em nome da sociedade, a gente agradece todos que estão organizando mais esse evento, e a gente deseja para todos os associados e os não associados que queiram se associar. É importante porque vocês, isso aqui, o suporte que a sociedade dá para esses eventos depende da mensalidade do sócio. Isso é uma fonte de renda nossa, e volta para vocês dessa forma, uma forma de eventos online, gratuitos, e só é possível quando se tem o suporte de alguém, e a sociedade tem dado o apoio a todos esses eventos. Então, desejo a todos um bom evento, e agradeço novamente a organização do SES por organizar mais esse evento Magnífico. Obrigado.
Obrigado, Danilo. Rafael, com você.
Obrigado, Gustavo. Bom dia, pessoal. É uma satisfação especial para mim, assim, estar hoje aqui com vocês, porque eu estava lembrando, Gustavo, do primeiro SES, que foi feito lá em Presidente Prudente, e assim, ao longo desse tempo a gente pode, pelo menos eu pude perceber o quão esse evento foi ficando maduro ao longo do tempo. Na primeira, a gente tinha algo ao redor de 50 pessoas, era um grupo pequeno, e hoje nós temos, nesse momento, 489 inscritos. Então, é uma satisfação especial ver essa história toda, né, do Simpósio de Ectologia Sistêmica. E é bom ver que a participação vem aumentando ano após ano, né? Então, eu, assim, em nome da comissão organizadora, assim, eu já dou meus parabéns, não para a comissão, mas para todas essas pessoas que estão aqui assistindo a gente, né? Isso é prestigiar a fisiologia, prestigiar a sociedade, é prestigiar essa área que a gente tanto gosta. Só o Gustavo, o editor-chefe da revista, me pediu para que eu falasse algumas palavras sobre a revista, então eu gostaria só de lembrá-los que talvez uma das principais bandeiras da nossa sociedade seja a revista de fisiologia, né? E essa bandeira é uma bandeira que a gente tem que carregar, né, junto com o editor. Então, de antemão, eu gostaria de convidá-los, vocês que estão assistindo, os próprios palestrantes que vão fazer parte desse evento, que pensem na possibilidade de enviar os seus trabalhos para a revista de fisiologia, tá bom? O professor Lüttge vai receber um convite especial, eu já sei disso, o passarinho verde já me contou, e eu espero que a gente mantenha essa tradição de sempre, após um SES, a gente tenha isso cristalizado num texto publicado em nossa revista, né? Um texto referente à programação do evento. Então, sem mais, viu, Gustavo, eu agradeço e desejo sucesso para esses próximos 3 dias de apresentações e de belíssimas palestras, tá bom? Obrigado.
Obrigado a você, Rafael. Bom, eu acho que a abertura desse quarto evento, né, justamente nessa época que a gente está vivendo, não poderia ser melhor, né, do que com uma palestra dos ícones, né, pelo menos eu considero, da ecofisiologia, professor Ulrich Lüttge. Professor Ulrich Lüttge é professor emérito da Universidade Técnica de Darmstadt, né. Professor Ulrich Lüttge tem trabalhado por mais de 50 anos, né, em diversas áreas da fisiologia, desde biofísica de transporte de membrana, até ecofisiologia de espécies tropicais, passando por ecofisiologia de fotossíntese, especialmente de espécies CAN, né, e o professor Ulrich também tem uma grande contribuição dentro dos estudos teóricos para a ecofisiologia sistêmica, né, discutindo questões relacionadas a uma perspectiva de Gaia dentro do pensamento ecossistêmico, né, bem como a biologia teórica, né? Então, trabalhando por temas como memória, dinâmica não linear. Então, eu acho que o professor Ulrich, ele representa, ele sumariza tudo que o SES e as pessoas que estão envolvidas com ele. Então, é uma grande honra, né, nós termos ele aqui. Ulrich, I'd like to thank you so much for accepting our invitation. It's a great honor. It's a great joy having you here with us. And you can please start whenever you want your presentation. Thank you so much, Ulrich, again.
Obrigado, muy obrigado, Gustavo, for the very nice introduction. I understand the technical terms which you used in Portuguese. Is that now size enough, or
it's okay. It's okay.
It's okay like that?
Yeah, it's okay like that. You can start your presentation whenever you want.
I'm very grateful for the invitation to my very first broadcast talk. Normally I talk to people in audience, and my first slide is simply introductory slide, and you see here on the right side the historical gate of the botanical garden in Darmstadt where I work. And on the left side, you see a beautiful picture by the famous Impressionist, French Impressionist painter, Claude Monet. And it is also a botanical garden. It is the Jardin de Giverny. And I will tell you in a few moments more about this botanical garden. But first of all, I want to come up with a provocation against all our efforts in biological studies. It is from the neurologist and philosopher Victor von Weizsäcker, who lectured in about exactly 100 years ago, in fact. And he said, with each successful step towards understanding mechanisms, biology diverts more and more from understanding life. And this is a fundamental intrinsic contradiction within biology and its tragedy as a science. And you may agree with me that for us biologists, this is a provocation. And this afternoon, today, I want to look at emergence, integrative emergence. And at the end, I will show you that we can find arguments against this idea from Weizsäcker, which is mainly based on the totally mechanistic and materialist way of some of biology, but we will do better when we look at these things, which I want to show you today in my presentation. So my layout is that, first of all, I want to explain the terms modularity and emergence. What are we dealing here with? And then I want to come to an older approach to systems biology. In fact, already in the 1970s and 80s, in plant biology, there was a lot of work on whole plant physiology, mainly root-shoot interactions, and integrations. And we can see that this is systems biology, long time ago. And of course, for that, we need signaling, and I want to talk about signaling in the systems, but also in ecology, or as you would put it, in systems ecology. And then we can wind up and come to a point showing that memory is a very important aspect in the relation to emergence. And then at the end, I will show what I said, that we derive very new views on systems biology from considering these relations and terms. So let's start then with the modularity and emergence, and I will come up with another provocation. Plants are by many people considered as modular organisms, and they have semi-independent or even totally independent parts. And then it comes to the point that these people say the performance of whole plants is a byproduct of responses of the modules. And the provocation, of course, by Haukioja is a tree is not a tightly integrated organism, but a byproduct of its parts. And we will go through it. You can see the modules, of course, as stems and branches and leaves and so on and so forth. These are the modules. And these are what he thinks the tree is only a byproduct of that. We will see about that when we go on. And from that, we can come to the next point, which is quite different, and it is now emergence. And this is this garden, the Jardin de Giverny, by Claude Monet, and we owe this very beautiful metaphor, the physicist Robert Loughlin. He said that this is emergent from modules. And you can imagine there is a painter, and he has a lot of buckets with paint and a brush, and he puts the brush in the paint and throws a patch of paint to the canvas. And then he does this repeatedly with the different buckets and the different paints and so on and so forth. And it is clear that everybody from you could also do this very easily. But would you arrive at such a beautiful picture? And you can see that from the integration of these modules, such patches of paint, you arrive with emergence of such a beautiful picture. And I can tell you that I think that emergence has a great aesthetic appeal, and there is poetry in it. So we have determined these terms, but now we can go to the molecular level. And I give you an example, another example at the molecular level. You can look at the proteome, and you have a electrophoresis or a thin layer chromatography and so on, and you separated all these proteins, and here you have the proteome of your plant or your cell or whatever you have. And these are all the modules. And the modules don't tell you anything. They're only black dots, right? But the modules are there. And now some of them can self-organize. They get together and self-organize and make a totally new structure, like you have an example here on the right. This example is a proton transporting ATPase at the tonoplast of plant cells, pumping protons from the cytosol to the vacuole on the expense of ATP. And it is built up of all these protein units. Some of those, you don't realize them here, but suddenly from the self-organization, you get the emergence and something very, very new, which is not inherent in the individual properties of these modules, has been formed and functions beautiful and doing this basically physiologically very important process of proton transport across the tonoplast membrane. So you can look at this at a large range of scalar levels. Let's say I have just talked about proteins here, but we can talk about a single molecule. And a single molecule has atomic particles. Let's think about a chlorophyll molecule. And you have electrons. And the electrons can be lifted up to a higher energy level, and there's a different energy, and then the chlorophyll is involved in photosynthesis and so on. But you have atomic particles in a chlorophyll molecule. And what is emergent is something new in the chlorophyll molecule. And then can go on through the levels. A membrane has various proteins, but the property of the membrane, and also lipid molecules, of course, is quite different from the property of the individual molecules. And so on. I abbreviate it because by now you will understand what I'm talking about. A membrane is making up a cell with its compartments, again emerging something new. Cells are making up tissues. Tissues are making up organs. Organs are making up whole plants. And whole plants make up all ecosystems. So you have in a large range, and this is depicted here, of scalar levels. If you look at down here at atomic particles, this is down here, but all the other levels, interestingly, in this double logarithmic plot, are forming a straight line. And you have the molecules here and so on. You have the biomes and the ecosystems. Here you have the whole globe, the whole biosphere. And you're going through these hierarchical levels. And now we can go and look at this in a more theoretical way when we look at networks. So let's say a blue circle here is a module. Well, whatever it is a module, and with other, it makes a network. But we can also say, okay, may all the blue modules and its entire network are condensed into another module. So we arrive at the brown module. And once again, these brown modules can form a network on the next higher scalar level. And of course, they can also be condensed again to a single module, which is a green module, and so on and so on. And you have a theoretical explanation of this. And it is like the Russian dolls, of course. And here is a metabolic example. You have a cell with a mitochondrion for respiration. All the individual enzyme reactions here are modules. But then this is a module on the next scalar level, and so on. And here you chloroplast and the metabolism in the cytoplasm. So you get the hierarchy of networks. And by now, I think we have seen enough, I have talked enough to arrive already at a first summary. So that will give us a guide as I go along in this talk. We have looked at this here, the proteins and the ATPase as an example, and we see that the modules are repetitive units, whereas this is a unique unitary system. And this is built up by self-organization. If you dismantle it, you could arrive to the individual modules, you have a separation. And one way is the holism, and the opposite way is the reductionism. This is made up by the integration of the components, of course, or by the isolation of the components in the other way. And this is, of course, unfolding complexity here after the emerging, whereas the other one is specialization. You look only at one particular item. It may be very interesting, and it is also not to be forgotten that we do need the structure and the properties and all the identities of the modules. We need to study this. It is just not the whole story. So we have specialization here and generalization on the other side. So the rest of the talk will go always through this, and you can bear it in mind, so that you always can think of where I am at a particular moment during the presentation. I come back to the physicist Loveland. He says that any laws of physics are emergent. They're emergent on a higher level, organizational level, from the laws on the lower level. A simple example, but a very instructive example, is the general gas equation. You know, of course, we are sitting here in a room with a gas, and the gas has a temperature and it has a pressure. But an individual gas molecule has neither a temperature nor a pressure. So this is emergent in the gas. And you can, if you prefer to look at not at a gas, but at a plant cell, we have a buildup of turgor. Of course, they have osmotic processes. You have solvents and solute molecules, and they can build up a pressure. And we know it, of course, in plant cells as the turgor pressure. Again, a simple molecule doesn't have a pressure. Only emerging from the integration of all these molecules in such an osmotic system, we arrive at a pressure. Example of the ATPase I have already shown here on the molecular level. This is a different one. It is the proton transporting was before in the vacuole, and this is the ATP synthesizing ATPase of the thylakoid membranes and the chloroplasts, which transport protons and make ATP. We all have another example here. Ribulose bisphosphate carboxylase oxygenase is a multi-subunit enzyme, and one very important intermediary metabolism enzyme is pyruvate decarboxylase, which splits the pyruvate into a CO2 molecule here and an acetyl residue. And this is an extremely complicated multi-enzyme system with very many different steps. I'm not necessary to go through all the details. It's just a principle that we need to see. And of course, if you look at biochemistry books, you can find out a plethora of examples. It's just a few I have arbitrarily chosen here to show you the principle. Let's go to a higher level, ecological level. You have a forest, and the forest is composed of trees, but the forest has extremely different properties than an individual tree. We even have the proverb in German, maybe you have it also in Portuguese, that one doesn't see the forest for as many trees. So that would be an ecosystem level example. And with this, we can start and generalize. And a very useful term for this is the term of the Holobjotan. What is a holobjotan? The original definition of a holobjotan is that it's a eukaryotic system, a eukaryotic organism, together with its kind of secondary often monocellular members and prokaryotic organisms, bacterial cells, and so on. We are, all of us, carrying about a kilogram of bacteria with us, and these are 10 to 100 bacterial cells per body cell, and it is 100 to 150 fold the number of genes altogether, which are then our own genes. So we are, in this definition, a holobjotan. And now we have worked on this concept, and we have extended the definition from dekaryotic host plus prokaryotic symbionts or other cells involved, to general. We can say generally it is a system where a different kind of organisms get together. The unicellular example here is a zoological heterotrophic cell, Paramecium. And this cell has swallowed a lot of chlorella cells, and it doesn't digest them, but keeps them, and so it is able to make photosynthesis. It is a holobjotan because it is the two cells together. That is on the more or less unicellular level, and you can go on through all these levels of all these scalar levels of organization I have talked about, and then you can see what I mean. You can come to ecosystems, you can come to biomes, and you can eventually wind up with the whole biosphere, or as James Lovelock would call it, the Gaia, the Greek goddess of Gaia is the whole biosphere. So you have again the scalar levels, and you can see what I mean each time it is emergent. Of course, the whole biosphere is different from man, and man is different from all its bacteria and so on. So again, we have emergence of extremely new properties. So having talked about this, I come to this example of systems biology, talking about whole plant physiology in the 1970s and 1980s. The system is clear. We have a leaf and we have a root, and they are connected by a shoot, and there are communications, in this case, phloem and xylem, and many different things are communicated and shared between these systems. And you can see what I mean when I say it is only the isolation of the modules, as it was originally in my example with the tree, and only a byproduct. It is the integration of these modules, which we can see. And of course, there is mineral and water from root to shoot, and also back from shoot to root. Photosynthesis products are sent from the shoot to the root via the phloem, and we have all these signals. Phytohormones from leaf to root, and electric, hydraulic, chemical signals, also other phytohormones from root to leaves. And you can see already that it is again an emergent whole plant system. All these modules are integrated in the self-organization of this entire plant. One of the pioneers of whole plant physiology in the 1970s was James Sutcliffe. And he was studying very much little young seedlings. And he was also very much interested when they grow, how the reserves from the seeds are mobilized. And one example of all his work is mobilization of the mineral, of the constituents. And on top, we have the element in the enumerator is mobilized faster. And on the bottom, the element of the denominator is mobilized more rapidly. So you can see that the building systems like carbon and nitrogen are mobilized faster than the minerals, except for potassium. Potassium is mobilized faster than nitrogen and carbon. And we know, of course, that potassium is very important in cell physiology. So all the reactions which are needed now to build up these young seedlings need a lot of potassium to have all the functions stabilized. The next example is the reduction of nitrate. If we give a root nitrate, it can easily reduce the nitrate at low concentrations. But if the root is getting more nitrate, it is not performing very well. It is not able to cope. It is the leaves which are in business for the greater part of the reduction of the nitrogen. This, of course, is due The roots have leucoplasts, organelles in which nitrogen reduction can occur, but only to a limited case because of energy reasons, whereas in the leaf and the chloroplasts, the photosynthetic energy, electron transport, ATP formation, and so on and so forth, is very much supporting, of course, the reduction of nitrate, and we can see this is working very well. So we have this interaction again. It's a specific example of the original of this slide where I showed all the basic principles. This is simply a specific example of it. Another example is salinity. If you have a ricinus plant in a saline environment, it will take up a lot of sodium chloride. And this is, of course, quite poisonous. Sodium ions are very poisonous in cells when they accumulate. So the brown squares are accumulation. It's large in the root, and it's increasing slower as you move up the plant to the younger leaves and internodes. And some of the sodium chloride is taken up, as you can see with the red arrows here, but this is remaining in the lower part and even recycled back. Blue arrows. Sodium chloride is recycled back to the ground. And you can see this is limited to the lower part, and the upper part, where the things are still very active metabolically and still growing and so on, are saved from this load of sodium chloride. It's another example of the whole plant integration of functions, and far from the individual modules simply telling the whole story. So with this, I come to the next point, because you have seen, of course, already, and I will illustrate a little bit with some examples. We need signaling within the systems, and we have different types of signals. One group of signals are electrical signals. We have action potentials, which are very fast. I put this in bracket because this is a point in literature which I'm not really very 100% sure if that's all right. It's very, very fast, but this is still quite fast. It's not as fast as our human electrical signals, but it is all right. And there are variations potentials which are slower, and system potentials also. The action potentials are rising from negative membrane potential rising to close to zero, small positive values, and then a recovery, there's a refractory state, and then it can be stimulated again. This is associated with a calcium wave, and of course the functions are changing conductivities for chloride and also for potassium, GCl and GK, and the associated fluxes of potassium and chloride make up these signals. It is, of course, necessary that we can show that these signals are transported in the plants. One simple system, as everybody knows, is a sensitive plant, mimosa. If you excite it and you have an external electrode, you can put here on the leaf or inside the tissue, press a little bit inside the tissue, and here in the petiole, and you have excitation, then a potential runs from this electrode to that, and you can record it here with your recorder. Technically, it is quite difficult to do this in a plant tissue because as you see, you need two electrodes to insert. And this is technically often a problem. You have one inserted and you try to insert the second one and off goes the first one. So manipulation is hard. It has been done and this is successful, but we used a little trick. Here you have a green leaf of Önotera. And if you put the electrode here in one of the cells, which is not so difficult, and the other one is in the reference, in the bathing solution, you get a light-dark signal. When you switch off photosynthesis, the ionic balances in the cells are changing, and this is seen in terms of a different potential. It is a transient development of the electrical potential, which you can see. Depolarization first, hyperpolarization first, and then coming back. And if you switch on the light, in other words, you go back from respiration to photosynthesis, you have the signal, but it is in the reverse direction. This is can be explained on the biophysical level, but it doesn't matter here. The only thing matters is when we have the electrode in the green cell, we get the signal. So these leaves of Inotera have mutated leaves that are not parts which are not green. And here is a part where the whole section, the whole cross section of the leaf is not green. Put electrode in, like here, switch off light and dark, change between light and dark, and you see no signal. So it's clear that only green cells can generate a signal. The yellow cells, the mutated chloroplastids, cannot generate a signal. So these are the controls. Now we put the electrode again here, and we wouldn't get a signal like here. But down here, you look, there is a little layer of green cells. In this part of the variegated Inotera leaf, we have one layer of green cells, and they can, of course, generate a signal because they're green. And now we do the light-dark switch, and lo and behold, we get beautiful signal in the cell, which wouldn't by itself show it, but has it got here, and you can see same signal as above, and there is no delay. It's very, very rapid. And you can see the only explanation, of course, is that the signal has been transported from the green cells to the electrode here in one of the yellow cells. So that is how we were able to show the transport of the electrical signals. Another type of signals is the hormonal signals. One of the most important hormones is indole acetic acid, and it is transported polarly in plant tissues because you have indole acetic acid transporters only at the lower, if you can take this as a coleoptile, for instance, or a shoot or so, at the lower end of cells. At the other end don't have these transporters. And so it is clear that the transport is more heavy here and is more heavy here for the electrically charged indole acetic acid. It's an anion. But the concentration gradient is maintained by proton transport. ATPase makes the cell wall compartment acid, and we can see then we get a non-dissociated indole acetic acid. But when as soon as it is in the cytoplasm, pH is alkaline, little 7.5, and you can see that it is then protonated, and that keeps the gradient between the protonated form, which is deprotonated at a more alkaline pH, and then transported again here and so on. So this is a mechanism of polar cellular transport of indole acetic acid. And here you can see the example in a developing Arabidopsis leaf. The hydatoid tissue has formed a lot of indole acetic acid, and that simply diffuses then in this particular transport mechanism into the leaf, and you has a direction of diffusion. You can see it elicits the formation of veins, of secondary veins, and of course a lot of hydatoids along the leaf, and then the whole venation of the growing leaf is regulated. So again, you have this function. And another example of hormones is the gravitropism. If we have a root in a normal orthogonal position, there is indole acetic acid transport from shoot to root. The indole acetic acid is distributed evenly, symmetrically in the calyptra, in the root tip, and it is transported back in the cortex, and this is quite symmetric. If we now turn the root, this changes. The back transport of indole acetic acid is much more heavy at the now physically lower side. And we can get here more transport. In other words, also the local concentration of indole acetic acid is higher here. And together with this, we also view electrical polarization. Here the cells are symmetric, minus 118 millivolt membrane potential on both sides. If we turn the root, it is changing. It's minus 131 here and minus 93 membrane potential down here. So we have both the gradient of indole acetic acid and electrical gradient. One doesn't even know exactly which is timing, which is first, hen and egg problem, a typical hen and egg problem. Both are observed together. And an additional hormone is cytokinin, which is also formed in the root tip and transported back. And together, these hormonal gradients then generate a response of the plant, which is a bending down, and it is a positive gravitropic reaction. And you have again a wonderful example of regulation by signaling and corresponding responses in the whole plant system. Another example which has been used very much in plant physiology is the split root system. You plant a plant so that roots can develop in two separate chambers. They're quite separated here, but roots can develop here. It is also called fence sitter, because imagine you are sitting on a fence and one of your legs you put to the left side and the other one you put to the right side, and you are sitting on the fence. And that's why we call it fence sitter plant or split root system. And now you can do very nice experiments with this. You can, in one chamber, have a very high water potential, a lot of water availability, low water potential in the other chamber. Now, we know that the water potential of the root growth soil, or in this case, the growth chambers, the water potential signal given to the shoot by two ways. One is in a chemical form, is abscisic acid, is a phytohormone, but also very important is the hydraulic signal. And so that arrives here. And the reason is that a leaf can start protecting the plant by closing the stomata before a water deficit is building up here, because it knows that the water deficit is going to come as the root system doesn't have enough water anymore. So this signaling is very useful for the whole plant performance. In this way, it is a kind of a bad or wrong message, because the plant doesn't have any water problems at all. Because by the other part of the root system here, it is in high water potential and can transport enough water to the leaves, so that would be not a problem. But nevertheless, the signal is arriving because the other part of the root system is developing these signals and transports it. So that is another nice example of whole plant performance in relation to signaling. And now we come to this other example, which is insect maybe coming to a leaf and tries to feed of it, and then of course the plant will react. The plant is forming chemicals, defense chemicals, and different kinds of things, also maybe things which don't taste very well to the insect and so on, and then the insect is moving away. The plant can also signal that this has happened to other parts. For instance, with another hormone, jasmonic acid is a very important stress hormone involved in such herbivoric interactions. And you can see it needs to be transported out of the leaf and transported back in the shoot to other leaves. It arrives there, and then the plant will make the repellent substances, and the insect will be drawn off. That is a long process, and all the time needed for all this transport here. But the plant also can make organic compounds, signaling compounds, which are volatile. Volatile organic compounds. The plant makes this compound here. The compound is volatile. It goes through all the other parts of the plant and signals to this leaf, hello, there is some problem. Herbivore is there, and the leaf will make the defense chemicals. And when the herbivore arrives, it's already bad. It has to go off immediately. We come to the right-hand part in a moment when we talk about ecology. So you have some examples of this systemic signaling. And I only make a couple of examples on the ecological signaling. And for this, we can return to the previous slide. Of course, the signal, the volatile signal, is not only distributed in the same plant, it can also arrive at neighboring plants. And then the plant is already warned, and when the herbivore arrives, not good place for eating. There's even a nice study which I have made in the savannas in the tropics with the giraffes in Africa. The plants make this also again, not again only against insects or whatever, make it also against giraffes, of course. Giraffe is feeding on a plant, and some gaseous warning substances are produced, and also the plant starts to make substances which don't taste so very well to the giraffe. So that is a good defense, like we see it here in the picture. But the giraffe, of course, is a more evolved animal with a lot of cognitive capabilities. And it turns out that the giraffe is not going on to feed on the weather, on the windward side, because there the volatile signals will arrive. So the giraffe is moving to the leeward side, where the wind is coming from, and not has any accumulated, any volatile warning substances signals. And so the giraffe can happily feed on these plants on the other side from where the wind is coming. So these are not anecdotes, it has been studied, and this is quite a nice example. And come back to the fence sitter plant. What you can do also is, in one part, you have a lot of nutrients, and in the other part, you have not very many nutrients. What the plant then is doing, it develops a large root system where it is useful into the chamber where the nutrient concentration is high, and only a little bit of root in the other chamber where there is not enough nutrients anyway to gain. Now, if we plant competitors to this plant, we can do this, and we plant them in the chamber with a low nutrient. The competitors don't have a very nice life. They're quite sorry because nothing very much is to be gained here. Whereas the other plant, the original host plant, has its root system in the chamber with a lot of nutrients and doesn't have any problems. And we can see this is territoriality of roots, which is again regulated. And the result is given here. The fence sitter plant has a lot of fruits and seeds and is very productive, whereas the competitor, as the number of competing plants is increasing, is doing increasingly bad. So that is another example of signaling in a systemic, ecologicalism systemic system. So we have arrived here, and now I make a big jump, but it will be seen that memory is a very important aspect of the emergence and of the whole plant performance. And I want to give you a little bit of an idea about some interesting studies of plant memory. So we click the wrong one. This is here, and now we come here. Okay, I am with you again. Here is memory. And first of all, we can generalize. I think it is fair to say that all living organisms which have biomembranes, polynucleotides, and proteins can develop membranes. This is what we need for the basic organization of cells, be them very simple prokaryotic cells or be them complex eukaryotic cells. This is the basic in the things we have available there, and they can always develop memory. I'll show you examples. But first of all, a little overview. What can we distinguish in plants? We have two types. We have habituation, learning, or priming memory, more or less synonymous. There are again two ways. These kind of memories can have familiarization, which means harmless stimuli are remembered but are ignored. Let's say you have a plant in an open and there is a little bit of wind. There's a small breeze and the plants will be moved and there will be mechanical signals. But the plant shouldn't react to this. This is a normal situation. So it's harmless stimuli and they are simply ignored. But there can also be violent responses, violent signals. And then this we call sensitization. And the responses are increasingly violent when we have harmful stimuli. So this is the habituation, learning or priming memory. And a more complex way of memory is the store and recall memory. And I will talk a little bit more about that in a few moments. First of all, as a general statement is that we have hysteresis. There are many, many examples of hysteresis, and any kind of hysteresis, in fact, is memory. An example is photosynthesis here of a green cryptogram met on tropical Inselberg. I've studied a lot Inselbergs, also in Brazil, of course. And we increase light intensity, and then, of course, photosynthesis, CO2 exchange in this case is increasing like that. And we keep it here at the end at high light intensity. And then it may be that at this very high light intensity, the plant is getting under light stress. So there is an inhibition due to the light stress, photoinhibition. And now we go back to lower light intensities again, and we can see the plant is remembering that it has been up here. But in this higher light intensity, now it is stressed. And you have the hysteresis loop and coming down here. So it's kind of a memory because these two loops here are separated. A very nice example for unicellular organism is this example of glucose uptake by Tanner and his colleagues of chlorella. Chlorella normally doesn't take up glucose and doesn't metabolize the glucose because it is green and performs photosynthesis. And keep the chlorella in the darkness and give it glucose. That's the only way of getting energy and substrates. You can't take up the glucose. We can see at 0 hour here, we start giving glucose, 20 minutes, no glucose uptake. But now you can see gradually it learns, and it has learned to take up glucose, and there is glucose uptake. So that is an induction of glucose uptake by giving the glucose. How it works, I will tell you in a moment. And there is this uptake of the glucose. Now you remove the glucose again, and you leave the plant in the darkness. And after 5 hours, you give it glucose again. And you can see immediately it can take up glucose 5 hours. It has remembered that it had learned before, 5 hours earlier, how to take up glucose. We wait 10 hours, same thing. It remembers for 10 hours that before it has learned how to take up glucose. We do it after 13 hours, it has forgotten it. So it's a turnover. It's very important to note this aspect of turnover. We will see in a moment how important that is. So then it has forgotten again, and of course you cannot use it again, and up it comes. But first of all, after 13 hours, it was forgotten. This is very, very important Nobel Prize winning finding theory of regulator operator, of how genetic systems can work, by Jacot and Monod. And up here we have the scheme, a cartoon of the original hypothesis, and then it's of course a theory because it's well supported. It is they worked not with glucose uptake as before. They worked with galactoside uptake. Beta galactoside cannot be metabolism by bacteria Escherichia coli. But then, as before in the chlorella example, the cell can learn it. So how does it function? There is a regulator gene, and this regulator gene is coding for the formation of a repressor. It can make different kinds of repressor for different genes. So it can make an active repressor which would fit to the operator gene. And when it has bound to the operator gene, the transcription cannot occur anymore, the gene is blocked. And alternatively, other possibilities is that it makes an inactive repressor, but then an effector can bind to the inactive repressor and then makes the repressor active, and then it can you can move to the operator and block the operator, and you can see that now again the gene cannot work. So the external signals are these effectors. Signal is probably leading to the formation of a receptor and then an effector, and then of course the repressor can bind it and change like that also. So you can see again we have a regulation on the molecular level, and you can see why after some time it is forgotten, because the system of course has a turnover time, and when the signal is not working anymore, then after a little while, 13 hours in the case of glucose uptake of chlorella, it is not functioning anymore. That was example from the prokaryotes. And in eukaryotes, of course, we have a different organization of DNA and chromosomes and the histones and so on, which is depicted here. We have the black line is the DNA, and the brown circles are the histones. And now we come to what I have shown you in the beginning, and I will explain later, the epigenetic memory. If the histone molecules, and also the black dots are here, the cytosine residues on the DNA are acetylated. If this is acetyl, then this is a rather large molecule, and it keeps the histones apart. So there is some distance between them, and there is enough room for molecules to enter and go and add to the promoter genes, so that the promoter genes can be regulated. If these molecules, histones and also the cytosine residues of DNA are methylated, methyl is a much smaller molecule, and then the histones are closer together, and there is no excess anymore, no way of excess, and then the gene is stopped. The gene cannot be transcribed anymore, and the gene is locked down. So this is epigenesis, and the point is now that methylation can occur in response to changing environmental conditions. The equilibrium between acetylation and methylation can change in reaction to environmental conditions, and these can be remembered, and in some cases it has been shown that they can even be inherited to progeny. So you have epigenetic involvement in memory. And I forgot to say, but it is important to say that no change at all during this whole process of methylation, acetylation of the sequence of the bases in the DNA. The DNA is totally the same as before, but the access, of course, to transcription is regulated by methylation, demethylation. So from this, we can build the epigenetic memory. We have stimuli cue environmental cues, signals are generated, and then the histone is modified, and we can have methylation, demethylation on the modified chromatin, histones, and also chromatin is modified. The mark, or the epigenetic mark, is changing, but not the DNA sequence. And then we get different methylation status and the gene expression. Small RNRs are even involved in feedback to the system, so that there's also a feedback loop around here. So this was basically, and now I want to come to the story call memory, which, as I said, is a very specific system. My friend Michel Tellier and Odile Despier in France have studied a lot of this in plants. It's in a little book which I published in French and in English, and here is the quotation. I have translated this book into German. So examples. I will give 3 examples of reactions. Hypocotyl elongation, symmetry of bud growth, and production of meristem, of epidermis meristems. The stimuli can be various: mechanical stimuli touching, pricking, wounding, drought, cooling, cold shock, manipulation, and the manipulation of the plants, chemicals, and so on and so forth. So there are a lot of signals which can be used and can be shown that they are active and involved in this store recall memory. First example, it is hypercall elongation. And now we have the terms store and recall. In other words, we need two things. We need a stimulus or a signal, stimulus, which is the store. The stimulus information has to be stored, right? And then we need a recall function, which is a condition which allows that the stimulus is recalled, right? So these two we have in the following examples, we have to distinguish. And in this case, it was a pricking, mechanical pricking, let's say by a tip of a needle on day 6. And the plants were in nutrient medium, and nothing happens. That is, elongation rate is normal. Why? There's no recall function. It's only the store function. We know that the reaction is only occurring in water. In nutrient medium, they don't react, but in water, they react. And we can see this here when the stimulus is occurring while the plants have been transported to water. That is 25% inhibition of the hypercausal elongation. So the store function is on. It is in, the store function is made on by the arrow, but the recall function is already on. And of course, we have immediate reaction, right? There's a second control. And the experiment showing memory is here. If you make again like here in the nutrient, but not in the water, of course, like in the first control, no reaction. Look, now we transfer to water, and this activates the recall function, and immediately we have inhibition. So on day 6 or whatever this was, it was stimulated, but 2 days later it was reacting when the recall function was switched on. So you have clear demonstration of this memory and how the two things like store and recall are involved. My favorite one is in fact this breaking the symmetry of bud growth. They worked a lot with these bee-dense plants. These have opposite cotyledons, and in each situation here we have a bud, and so symmetric the buds, and you know, of course, they have so-called apical dominance. As long as you have a top bud, the main shoot will grow, and the lateral buds will not grow out. If you cut it, the lateral buds will grow out. And there are different ways. Mostly, both of them will grow out, but then one of them will win and will be leading. But we cannot say. It is totally symmetric. Both can grow out, and one of them will win and will lead and establish again apical dominance. But we cannot predict which one. It's totally symmetric. Now, if you prick one of the cotyledons, again with a needle, mechanical stimulus in this case, we have a signal, and the signal is transported, of course, here to the knot of the shoot with the buds and the axils of these cotyledons. And everybody knows, or you will know this maybe also, that the bud opposite to the changed to the signal, to the pricking, will gain. So either this one will grow out first or it will grow out faster. At least this opposite, but in high percentage, statistically significantly, will gain. After you cut off the top and remove the apical dominance. You can do this at the same time of pricking, and now you will not see any memory. But you can do this later. If you prick here and do this 2 weeks later, you cut the top bud. And only then you generate, this is of course the storage function again, and cutting the top bud is activating the recall function. Only then you will come, and it will take 2 weeks. Doesn't matter. It also doesn't matter which one you do first. If you cut first and then prick, or vice versa, as it is shown in the picture. So the store and the recall functions are quite independent of each other. And the storage is not reversible. If you have, once you have done this, you can prick the other side, the other correlator, and it will not have any effect. But what is also very interesting is, if you don't wait 2 weeks, here we waited 2 weeks to show the long time of memory. If you only wait 2 hours, then the removal of the store, the activation of the recall function will be only active if we do it after 2 days and not after 2 weeks. And we can see that in a way the endogenous clock, the diurnal rhythm, must be involved in the things, because after 2 days, breaking of the symmetry by cutting the top bud will only be active if we do it in the morning and not at midday. So you can see that the circadian rhythm, the day-night rhythm of the plants, is somehow involved. And that brings us to the biological clock. There will be 2 or 3 other talks to explain the biological clock. It's a very complicated system. We have morning genes, which are accumulating during the night and most active in the morning, like here. And we have evening genes, vice versa, accumulating during the night. And then we have the clock. And of course, you realize a clock in itself has nothing to do with memory. But a clock immediately gets memory if you have set points. A very simple and familiar example is your alarm clock. If you set your alarm clock, the clock will remember the time you have set and will give you the alarm at that particular point. So the clock can quite easily be involved in memory. And this is also why the phase can be changed. We can put the signal, of course, the set point. We can put the set point at a different position. And of course, that means change the phase. And one looks now with great interest at genes in plants which have phase information. So the clock is also involved. But coming back to the other examples, we can see the last example here, which is meristem formation. Again, as before, we have the 2 elements, stimulus, signal. In this case, it was transferred to new medium. I have said that handling the plants is a signal. If you take a plant out of the growth medium and put it into another growth medium without obviously disturbing it very much, it is nevertheless disturbed. This manipulation is a signal for the plant. And you need to know this, of course, if you do experiments, that after you manipulate plants, you have to give them some rest period until you can do sensible experiments. So that will be the signal or stimulus in this case. And the recall function will be remove calcium. And now you can see here, if we have a signal and calcium is removed, there will be meristem formation. That's the first control. If we only have the signal, no meristem formation. If we only make the recall function, only remove the calcium but don't give a signal, no meristem function, right? These 2 things only show you that both are needed. We have seen this before, in fact. Now here, memory. You give the transfer, the manipulation signal, no memory formation. You remove the calcium, and immediately you have meristem formation, which means, in this case, for a couple of days, here is the time scale, for a couple of days, the signal was remembered, and immediately the reaction is coming when you activate the recall function. So, signal remembered. Vice versa, you have control medium, remove the calcium, but there's no signal, no stimulus, no meristem formation. A few days later, you give the signal, you have activated already a few days before the recall function, and you have meristem formation, okay? Recall function remember. So independently, they can remember either signal or the recall information. And the most beautiful thing in this experiment is that you can even do this repeatedly. You have the first example is here, stimulus and removal of calcium simultaneously, and you have the blue curve, it's meristem formation. After some while, it comes to saturation. We look at the increase here to this particular level of meristem. Then you do the same with the yellow plants, and off it goes, like here, but now you give it a calcium excess here, and immediately the meristem formation is stopped, and it will never reach this high level because, of course, due to the calcium excess, you have removed the recall function. And the violet curve with the diamonds is an interesting one. You do this as before, and you stop it here, but now you deplete once again the calcium, and off it goes again. Like here, it was inhibited, but here it goes off again. And you can see I have repeatedly activated the recall information in this example. So in a general way, we have the elements. It is a simplified model of the plant memory. We're still looking for theory. We need model to get involved in this. I can only advertise it. We have retired and we didn't find people to do this. If somebody is good in mathematical modeling, it would be beautiful to work with these elements which we have taken together here and to make a functional memory on silicon. Log genes and stimuli, environmental cues, generate signals, electric, hydraulic, chemical, RNA, and so on. And these will activate effectors, like in the Jacob-Monod hypothesis or theory, or the biological clock, or the epigenetic memory. And then the genes are unlocked. And then you have the habituation priming memory, which goes rather direct to processes of life. Also, the signals will play a role here, of course, moving when the genes are unlocked. The signals will come in here, and again, and you get this, but you have here the recall and gene, and store recall gene memory. The activated recall genes will make their gene products, and they will, together with the activated store genes, generate the store gene products, and you get the processes of life like growth, development, differentiation, and so on and so forth. So that is the involvement of memory, and you can see also in emergence, of course, because from the simpler parts, you get the emergent systems of highly integrated plants. I think the hour is over, and I am also very close to finishing. We can come to a final consideration of emergence and integration, and this new view on systems biology. My wonderful example from epigenetics is this one here. It is Linaria vulgaris humano. This is a very frequent weed in Germany, in fields. It has this bilateral thigomorphic symmetry. And if only one gene, the cycloidea gene, promoter gene, if this is methylated, and in other words, you have learned before, I repeat it up here, when the system is methylated, the gene is locked down. It isn't working anymore. Only one gene, without any change in the whole DNA. You don't get this kind of flower anymore. You get a radial symmetry of flower, and it is emergence of a totally new system with just this one gene. And that brings me to my other favorite example, which is the grand piano. And it shows you how important the emergence is and how important the self-organization is. And we can also come to genomics again. Look, everything is there. The keys are there. The strings are there, the body, architectural body of the piano is there, the pedals are there, everything is there. There's no music, it's nothing. In fact, it is really nothing. It depends on who is playing on it. And also the complexity of what is played will be different depending on who is playing it. let's say your former South American cohabitant Daniel Barenboim is one of the most famous pianists. If he is playing it, or if I would play it, some very, very different music would come out. I can tell you that. And you can see it is now important to note who is playing. All these modules by themselves don't do anything. And it is like here. If they are there, okay. But they don't make an ATPase unless they are integrated by self-organization. And Emergent is this complicated multi-protein system. And it is like that here. Or you can look at the genes. The wheat Arabidopsis has more genes than man. A little nematode and a fruit fly has almost as many genes as man. And we have only 300 genes different from the chimpanzee. But I keep maintaining that we are quite different from chimpanzees. And you can see it is not the genome just producing the modules. It depends on how it is read and on how all these things are integrated, of what you may get out of it. So that brings us back to the very provocative statement of Victor von Weizsäcker. What we have seen in the talk is stimulus. There is information inherent in them. There is a storage of information in memory. There is self-organization as a category of holism and the integration of the plant. All this makes a unitary, unique system. And the answer to von Weizsäcker was, we are not just materialists. Without leaving the strict rules of natural science, which look materialistic, but without leaving them looking at these integrations, at these emergence, at these highly complex systems, without losing the sight of the different spiritual qualities of life, we can start and explain life. So you may forget the entire talk. Just don't forget the last picture. A rose is both. A rose is a biological system, a complex, integrated, emergent plant biological system. You can, with all your ways of studying plants, you can go and study its behavior and its comportment in ecosystem and whatever. And you do pure science. But you shouldn't forget, at the same time, a rose is a flower of overwhelming beauty. With this, I thank you for your attention, and I give you a few references. I don't hear you.
Oh, sorry.
I don't hear you. I haven't heard anything. I haven't heard anything.
My microphone is turned on.
I wondered why everything is quiet. I thought you may have gone to the beach.
Sorry so much. I just thank you so much. It was a beautiful introduction for our meeting. You cover many aspects that you'll be talking during this week. So thank you again. It's amazing presentation. Couldn't be better. We have some time to a few questions.
Right.
We have a question from, you put the question in the screen. Can you read it? The question from Daniel Santa Cruz Daminelli.
So I can go back to the slide. Can I go back to the slide?
I think your presentation is closed. Now.
Shall I open it? We don't have it anymore.
Yeah. But maybe you can just try to answer talking.
Well, of course, one doesn't know where it is stored. I mean, the molecular. This is a phenomenon which I have shown, that you can have storage and recall, but they have not done the molecular experiments to dissect. I mean, that would be a very, very difficult study, of course, and a study alone. But they were interested in showing a lot of different examples of that storage and recall is occurring. So there will be still a lot of possible ways to continue these experiments and find out about these molecular details, where it is stored. I have no answer.
Okay, thank you. We have another question from Cristiano Ferrara. Can you read it?
Yeah, yes. Is it possible to work with interaction networks? Yeah, I mean, this is done all the time. I mean, many examples have shown that you can do this. I mean, you can grow a chlorella cell and a paramecium cell separate from each other, and then the chlorella cell would not be within the paramecium cell. The advantage the chlorella cell has from the paramecium cell is it is protected from herbivores and so on, and from diseases. And the paramecium cell has the advantage it can do photosynthesis, but only if they merge. And you can easily do this. You have it separate cultures and then you mix the culture, and suddenly it happens that they get integrated and involved. Or the other one with the phytophagus in insect. You can, of course, do this in the field with the signaling, or with a camel. You can make these experiments. So I think you can do a lot of experiments.
Thank you.
Is that okay, or?
No, I think so. We have a few questions. Another one is from Daniel Santa Cruz, but it's Maria Teresa Portes that is asking you.
The calcium waves are a different topic in plant signaling and regulation, and it is said we have calcium in the vacuoles. A lot of calcium is stored in the vacuole, and also we have, of course, calcium in the external medium. And we know that there is very many experiments. I mean, calcium that would fill libraries of examples where calcium is regulating processes, and there is it is a guanosine system also with GTP is involved, and there are membrane processes which regulate these calcium waves. And of course, normally the cytoplasmic calcium concentration is extremely low. It's very, very low, and that is why calcium can be used as a signal molecule, a signal ion, because small increases from transport of calcium from the outside medium or from the vacuoles will change calcium concentrations. And in one experiment I have very short in the memory, I've shown very briefly only, there is a calcium wave, and these calcium waves are signals. And so the calcium physiology is a very large field, I think, in plant biology.
Okay, thank you, Ulrich. We have another question from Ana Luisa Sobral Paiva.
What is our main session? Aha! Yeah, this is that leave the question on while I respond to it. What can I answer?
Oh yeah, please.
It is a very good question. I think what we have in biology, in general, we have waves. We have waves where methodology makes a very, very strong progress. And then we have an increment of data accumulate. And then we get worried, what do we do with all those data? And then we get another wave, which is a wave of thinking. I give you, now I am old man, but I was very young, still as a scientist, when electron microscopy came up. And then people were starting to make electron micrographs. And at the beginning, we didn't understand them. We just saw things and membranes and particles and whatever. And there were only a very few number of experts who were able to say what we see. I can remember very easily that I didn't understand these electron micrographs until after a lot of, now I have interference of, shall I talk or?
Yeah.
I have a lot of interference on the phone.
No, but it's okay now.
So that was also first the technique and then all those information. And then at the beginning, I was not capable of interpreting electron micrographs. And then of course the thinking is coming. And the same we are having right now. We had this, that we derived all these techniques of genomics, transcriptomics, metabolomics, and whatever you call it. You call it proteomics, you can call it signalomics or channellomics or whatever you have. And you have mountains and mountains of data. And then you come to the idea, it is modules, which I showed also on this graph with the black dots with the molecules. You have a lot of information, but you don't know what it means. And then it comes to thinking. And I think it is true that we have now these techniques, and it is not so much a concern to get all this still exaggerated. Although, of course, we never should, it would be wrong to conclude that we should stop developing new techniques. But this point is that we need now two disciplines. One is bioinformatics, which is simply knowing how to handle this information. And the other one is biomathematics, which is develop theories of how to handle the genes and how to find the ideas which are behind all these informations of the modules of the omics. The omics are simply the models. And the new way, which is very important now in the bioinformatic sciences, is and biomathematics sciences especially, is the ideas. We need to develop ideas and find out what is behind all these building blocks, all these modules which we can depict. It's a very nice question. It is a philosophical, and maybe after some time we will learn how to do this, and then another wave will come with more techniques and again more thinking. I think these are rhythms which we go through. Electron microscopy is an example which is now more than 50 years old.
Great. We have more few questions. The next one is from André Parisi. This question is have two parts. Has two parts.
I don't know. I don't know how I can answer this, but I think this is maybe a little bit of a wrong question, because it is also specific, of course. I mean, not all modules can react to the same signal, because you have specific. As I said, a tree has modules of shoot and root and leaves and flowers and so on, but they will react on different signals. Root will react on water and nutrients, and the leaf will react on light. So I don't think that this is a very easy.
Okay, thank you. We have another question from Danilo.
No, not necessary. I don't think it's necessary. It is only a possibility. And in one of the slides where I had shown you that I can build a theoretical model of the memory, I had three terms, which one of them was epigenetics, but there were other ways also. It was when I said we need to challenge modelers to make. Sorry that I cannot show the slide again, but there were three examples, and epigenetic is just one.
Thank you, Ulrich. We have two questions. The first one from Paulo Mioto.
Aha. Yes, of course. I mean, you will have examples where the shock is a general problem. I mean, a cold shock is generating certain reactions, and you will have the same reaction. And there are other signals where they will not perform in a similar way. So it again is quite specific. I mean, all these downhill processes, of course, can then be quite specific. But in some cases, you may find examples where they behave for instance, wind. I mean, the different plant species will respond in similar ways to wind or gravity. These are stimuli, and of course, the plants perform in the same way to gravity.
We have another last question from Thiago.
I am not familiar with this. I know, of course, what you're talking about, but I am not familiar with using it, and I would not like to give an answer to that.
Okay, thank you, Ulrich. I think there is another question from the audience.
Okay.
No, I think don't. I think we have finished all the
covered
questions.
The questions.
No. I think that's it.
Okay.
Okay, the final one. Yeah, Rafael confirmed. Ulrich, so thank you again. I think it's a great beginning for our symposium. Could be better. And I am your reference to me, I think, for all my students. So it's a great joy to have you here. I could be more happy. So thank you a lot.
What should I do? I leave the studio now. Should I say leave the studio? Okay, all the best to you, and have a nice conference. Huh?
Thank you so much.
It's okay to press leave the studio, right?
It's okay. Thank you so much.
So,
bye. Bom, pessoal, a gente está terminando, então. Eu acho que foi uma grande introdução. Vocês vão perceber que ao longo das nossas outras sessões, né, a gente vai abordar vários aspectos que o professor Lüttge tratou. Então, foi realmente algo, acho que não poderia ser melhor, né? Acho que agora à tarde a gente vai ter a sessão sobre memória, inclusive com o nosso amigo aqui, Rafael, né, e às 5 horas. E então a gente espera que vocês, no próximo link, por favor, chequem a programação. Rafael, você tem mais alguma questão?
Não, foi maravilhosa essa palestra. Eu vejo vocês mais tarde, né? Nos encontramos mais tarde.
Isso, às 5 horas, então. Até mais, pessoal. Muito obrigado.